Randomized comparative experiments. The remedy for confounding is to perform a comparative experiment in which some individuals receive one treatment and similar units receive another. Most well-designed experiments compare two or more treatments. Some experiments may include a control group that receives an inactive treatment, placebo, or an existing baseline treatment. An experiment that uses both comparison of two or more treatments and random assignment of subjects to treatments is a randomized comparative experiment. Randomized comparative experiments. Comparison alone isn't enough. If the treatments are applied to groups that differ greatly, bias will result. The solution to the problem of bias is random assignment. In an experiment, random assignment means that individuals are assigned to treatments at random, that is, using some sort of chance process. Completely randomized design. In a completely randomized experimental design, all the subjects are allocated at random among all the treatments. Okay, so you start on the left of this flowchart with experimental units, which go to random assignment, and from then they're split off into group 1 and group 2. So if you follow group 1, they experience treatment 1, and group 2 experiences treatment 2, and then in the end, you compare all the results together. The logic of randomized comparative experiments. Random assignment of subjects forms groups that should be similar in all respects before the treatments are applied. A comparative experiment with randomization ensures that influences other than the experimental treatments operate equally on all groups. Differences in average response must be due either to the treatments or to the play of chance in the random assignment of subjects to the treatments. Principles of experimental design. The basic principles of statistical design of experiments are: control. Restrict the effects of lurking variables on the response, most simply by comparing two or more treatments. Randomization. Use chance to assign subjects to treatments. Replication. Use enough subjects in each group to reduce chance variation in the results. Statistical significance. An observed effect so large that it would rarely occur by chance is called statistically significant. A statistically significant association in data from a well-designed experiment does imply causation. Cautions about experimentation. The logic of a randomized comparative experiment depends on our ability to treat all the subjects the same in every way, except for the actual treatments being compared. Many medical experiments are, for example, placebo-controlled. A placebo is a dummy treatment that is as similar to the treatment as possible, but contains no active ingredient. Double-blind experiments. In a double-blind experiment, neither the subjects nor those who interact with them and measure the response variable know which treatment each subject is receiving. Control groups, placebos, and blinding. Control groups, handled identically to the treatment groups in all respects, except that they don't receive the actual treatment. Placebos, research shows that people also respond to placebos, looks like the real drug but has no active ingredients. Randomly assign some patients to receive the drug and others to receive a placebo without telling which they are receiving. Blinding, double-blind. Neither the participant nor the researcher taking measurements know who had which treatment. Single-blind. Only one of the two, participant or researcher, knows which treatment the participant was assigned. Example: blindly lowering cholesterol. Which lowers cholesterol more? Special diet, portfolio, versus drug, lovastatin. Details: 3 treatments, portfolio diet, low-fat diet with lovastatin, low-fat diet with placebo. The 46 volunteers were randomized by a statistician using a random number generator. Blinding. Researchers and participants, both blind as to which drug, lovastatin or placebo, people in those two groups were taking. However, participants and dietitians could not be blind to what the participants were eating. Lab staff evaluating cholesterol measurements were blinded to the treatment.